So the first molecule we're going to be going over is DNA, otherwise known as deoxyribonucleic acid. So like I said before, DNA is the genetic information in all organisms except for viruses that sometimes use RNA. So most organisms use DNA to store their genetic information, and that genetic code is used to make proteins that all our cells in our body use to function. So DNA is replicated and then also inherited from one generation of cells to another. Our DNA specifically is distributed among 46 chromosomes into 23 pairs. Other organisms will have a different number of chromosomes and different amount of DNA. But for humans, it's 46 chromosomes and 23 pairs. So DNA on each chromosome is made up of separate genes that transcribe into RNA and then finally translation into proteins. And DNA is double-stranded, unlike RNA, which we'll go over in the future. RNA can be either single-stranded or double-stranded. So the structure of DNA. So DNA is a polydeoxyribonucleotide. So that is made up of single monodeoxyribonucleotides. So on the right is the structure of a monodeoxyribonucleotide. So DNA uses deoxyribose as the 5-carbon pentose in the structure. So it's a ribose that instead of a 2' OH, it's replaced by a hydrogen instead. So so this right here was, instead of an OH, it was replaced by a hydrogen. So that's why it's called deoxyribose, while RNA is called ribonucleic acid because the OH is still present at that 2' position instead of being a hydrogen. And so the monodeoxyribonucleotide is made up of a nitrogenous base. There are 2 different kinds of nitrogenous bases. So the first is purines. Purines have 2 rings, and the 2 purines in DNA are adenine, the symbol for that is A, and guanine, which is G. So those are the 2 purines. They have 2 rings in their structure. And a mnemonic to remember that is pure as gold. So purines, pure, and then as gold. So the symbol for gold is Ag. So you can remember adenine and guanine. Ag is a purine because purine is pure as gold. So those are the purines. And then pyrimidines, those only have 1 ring, and it's cytosine, which is C, and then thymine, which is T in DNA. And then RNA, instead of thymine, they'll use uracil. So cytosine, thymine, and uracil are the pyrimidines. And then those only have 1 ring. So those are the 2 different kinds of nitrogenous bases. So what happens when you bind the pentose to the nitrogenous base? You get a nucleoside. So when you bind a 5-carbon pentose to the nitrogenous base, so the nitrogenous base is covalently linked to the 5-carbon pentose at the C1 position. And then on the right, I also have the structure of the 4 deoxyribonucleosides for DNA. So, and then finally, it forms a nucleotide when you join a nucleoside with either 1 or more phosphate groups attached at the C5 position of the carbon. So when you attach a phosphate group to a nucleoside, you get a nucleotide. And nucleotides have a high energy because of the negative charge repulsions of the phosphate groups. So phosphate groups are negative, and when you put two negative groups close to each other, they repel each other, and the negative charge repulsion is the reason that nucleotides have high energy. So molecules like ATP that you know, those are a nucleotide because so ATP, they have high energy because of the phosphate groups in the structure, and the negative charge repulsion generates that high energy. So there's the Watson and Crick model of DNA. So their experiments, they found that DNA is a double helix structure. So they have two linear chains wound in spiral. So like I said, DNA is double-stranded. So those two linear that double chain strand of two linear chain, the double chain strand of two linear chains are wound in spiral, and it forms a helix-looking structure. And the strands of DNA are anti-parallel. So one goes in the 5' to 3' position, and then the other strand goes in the 3' to 5' position. And so in the model, they found that the sugar and phosphate are on the outside of the helix, while the nitrogenous base points on the inside of the helix. And the model also shows that there's complementary base pairing. So the adenine binds to a thymine via 2 hydrogen bonds, while the guanine binds to a cytosine via 3 hydrogen bonds. So since the guanine and cytosine has 3 hydrogen bonds, that base pair is stronger than the adenine and thymine bond. And then the hydrogen bonds and hydrophobic interactions of DNA provides stability to the structure. So there's a rule called Chargaff's rule. So that's the rule that says that the total amount of purines has to be equal to the total amount of pyrimidines, which makes sense since a purine all binds with a pyrimidine. So adenine, which is a purine, binds with a thymine, which is a pyrimidine, and then guanine, which is also a purine, binds with cytosine, which is a pyrimidine. So since each base pair has one purine and one pyrimidine, the total amount of pyrimidines equals the amount of purines. So if you know the amount of purines or the amount of pyrimidines, you'll know the amount of the other. So, and then also all the total amount of adenines has to equal the total amount of thymines, while the total amount of guanine has to equal the total amount of cytosine, since they base pair with each other. So this is just a mini table that shows you how the total amount of purines and pyrimidines has to equal each other. So more into the structure of DNA. So DNA has a sugar phosphate backbone. So the sugar is made up of alternating the sugar phosphate backbone is made up of alternating sugars and phosphates. That's because the base pairs, the nucleotides are joined by phosphodiester bonds. So the 5' end of one molecule of nucleotide has the OH or phosphate bonded to C5. Sorry, this is wrong. It's supposed to be C3 of the sugar. No, sorry. Yeah, so the first nucleotide will have a 5' 5' end, and it will have a oxygen or phosphate bonded to that carbon. The carbon of the sugar. While the next nucleotide on the 3' end, it'll have a free OH on carbon number 3 of that sugar. And then these are the two parts that bond to each other through the phosphodiester bond. So it's a 5' to 3' phosphodiester bond. So the 5' phosphate of one sugar binds to the 3' oxygen OH of the next. So nucleotides are joined by 3' and 5' phosphodiester bonds, and like I said, the 3' carbon of one sugar bonds to the 5' phosphate of the next sugar. And then, so eukaryotic chromosomes, they organize the DNA into either heterochromatin or euchromatin. So heterochromatin, it's compact and transcriptionally silent. So that's DNA that isn't transcribed or replicated a lot. So when in interphase, you only have a small amount of it, because interphase, usually that's when the DNA is being used. So it'll be most likely euchromatin, which is dispersed chromatin. So that's genetically active chromatin. So this DNA, it's being transcribed a lot. They're making proteins from it. So this, as you can see in the diagram, euchromatin, it's less tightly bonded together. There's more space for molecules to interact with the DNA. While heterochromatin, they're compact, they're all bunched up together, so it's hard for them to be using the DNA, so that's why it's transcriptionally silent.